Hey guys, welcome to our first content lecture. So today we're going to be covering neuroanatomy, the first part of it. So let's get started. So the point of this diagram is to show kind of how the nervous system evolved over the years in different organisms. So in the beginning, it used to just be like a net of neurons, as you can see in the hydra. So there were signals that were propagated, but basically the hydra could only perform very simple functions. But then afterwards, these neurons clustered together, formed nerves, and eventually we got to something called cephalization. So cephalization is basically when we have neurons that are centralized in one location. So for us, it's the brain. And this is what really informs decision making and complex function. So this is like kind of the turning point in the development of nervous systems. So before I get started on the anatomy of a neuron, I just want to think about, let's say we had 2 points in our body, point A and point B, and we wanted to send a signal from point A to point B. Maybe our brain is telling us to move our hands or something like that. So what you might expect is that there's one long neuron that goes from point A to point B. Like if you didn't know anything, that's what I would expect. But instead, what our body does is it has a bunch of smaller neurons that kind of propagate, what we call it, the signal from point A to B. So having said that, let's take a look at the anatomy of the neuron. So first we have the soma, which is the cell body, and then that's surrounded by dendrites. So the way I like to think of dendrites are input points. So another neuron might be connecting here. So this would be its soma. And it would be connecting to these dendrites and sending a signal to it. And then afterwards, we have the axon hillock, which connects it to the axon. And the axon from one neuron connects to the dendrites of another neuron to propagate signals. And then something we have later on I'll talk about called glial cells. So they're not neurons, but they have very important supporting roles in neurons. So information is processed probably just as how you would expect it. So we have some kind of sensory input brought by sensory neurons. So maybe, I don't know, there's a spider on your hand. And then there's integration. So this happens in our brain. And sometimes it actually skips the brain and goes to our spinal cord. But I'll talk about that later. So our brain kind of processes what's happening. So you look at your hand and say, oh my God, there's a spider. So this is all happening because of interneurons, which are sending the signals back and forth in your brain. And then you get a motor output. So your brain says this spider is a threat. And then your motor output would be your hand kind of shakes around trying to shake the spider off. So the nervous system is composed of two main parts, the central nervous system and the peripheral nervous system. So the CNS, the central nervous system, consists of the brain and the spinal cord. So you can think of this as the intelligent part of your body. So this does all the thinking and all the integration and stuff like that. And then the peripheral nervous system would be, that carries information into and out of the CNS. So that would be something like the nerves on your hand that, send the signals to your brain, like what's going on. So the way it works in neuron cells is we have something called the sodium and potassium pump. So if you've taken bio, you already know about this. But basically, let's say this is our cell, say it's a circular cell, and this is a kind of zoomed in image of that. So the outside, this is the outside of the cell, and then this is the inside. So what happens is we get three sodium ions get pumped out for every 2 potassium ions that get pumped in. So if you do the math, you get a net positive charge that is exiting the cell, right? So the cell becomes slightly negative relative to the outside environment. But this actually only contributes, like if you think of it, a net of a +1 charge every time this like cycle runs through, right? This 3 sodiums go out and then 2 potassiums come in. But what actually happens is on the side, there's something called a potassium channel. So it's just constantly open and it allows potassium to flow freely. right? So if there's a lot of potassium here and not a lot of potassium here, potassium would flow this way, right? So then, and since potassium is positively charged in the cell, so we also have further positive flux out of the cell. So the cell becomes even more negative. So these are some important ion concentrations. It's not so important as to remember the numbers, but just remember that like at the end of the day, the cell becomes negative 70 millivolts charged relative to the outside environment, which is as I was saying before. This equation right here is something called the Nernst equation, and you can actually calculate exactly how much that is using that. But we won't go too much into that right now. So, like again, we have the neuron right here. That would be like the soma, and then this is like the axon. Might have some dendrites. Obviously, this is a very simplified drawing. But basically what happens is when the signal gets input into the dendrite, there are these channels that open, sodium channels. So if you imagine the cell, like this is the membrane, this is the inside, and this is the outside, then what happens is sodium comes in and starts, increasing, like in a depolarization, it starts increasing the charge inside the cell, right? And eventually, if this crosses a threshold, then we can get something called an action potential. So an action potential is like an all or nothing response. It doesn't matter if 100 sodiums flow in or 10 sodiums, as long as we're above this threshold, this action potential amount is going to get hit. And that's an important distinction I want to make. So let's say you want to close your hand, right? What dictates how hard you're going to close your hand? Right? Your intuition might tell you, hey, it might be the strength of how much your like neurons fire, right? But that's not true. Because an action potential, the principle is you can only fire at one strength, which is right here. So it actually dictates how strong your grip is how many times the neurons fire in unit time. So let's say it fires 10 times per second versus 100 times per second. That's what dictates the difference, right? The strength is always going to be the same. And this is important to remember, especially in the next lectures. We'll go on to neural encoding and decoding and that kind of stuff. It's important to remember the firing rate is actually the name of the game. That's what dictates everything. And then this right here is something called a hyperpolarization. So let's say an ion channel opened and instead the sodium flowed outwards. Then obviously the positive charge is leaving, so the inside becomes more negative. So this is the phase of the action potential. So first, there's the resting stage. Everything's just normal. And then we get a depolarization. So again, sodium's flowing inside. And as you can see, the charge is increasing. And then even more sodium channels open. It's kind of like a chain reaction. Even more sodium flows in. And then we reach the peak of the action potential. But then afterwards, it gets blocked by something called the inactivation loop. And see the difference between stage 4 and stage 1. Here is the channel is actually just closed. Here it's inactivated. And this is important for something I'm going to talk about next. Okay, and then this potassium leaves, so it becomes negative again. We undershoot it, and then it goes back to normal. So remember how I said here it's actually, in stage 4, it's actually inactivated, where here it's just closed. Well, since it's inactivated, that means no other action potentials can fire. So the graph won't go back up or something like that. That can't happen because these channels are inactivated, and that's called the refractory period. So this basically shows that an action potential can only travel one way along the axon. So here's our cell body again. Here's the axon. I won't draw the dendrites right now, but it can only travel this way. We don't want the action potential to travel the other way. So the way the body does this is it sends signals of sodium opening, to the right, I guess you could see in this diagram. So the signal can only travel one way along the axon and, can't go back to the soma. So if you've taken physics or anything like that, you know that if you want to conduct electricity, right, which is how all this is basically functioning since we're just exchanging positive and negative charge, right? So if you want to make electricity travel faster, right, then there's a couple things you can do. You can either cool it down, which we can't do in our system because, the body needs a certain temperature. So cooling down's out of the question. We could either make it really wide, increase the cross-sectional area. And, some organisms actually do this. Like the squid, they have a 1 millimeter long axon, which might not sound like much, but if you consider that there's like millions of neurons in us right now, that's huge, right? So you can do that, or you can just prevent charges from leaving the cell. So you don't have any like leaky areas of your axon. So when you take like a wire, right, it's surrounded by some insulating material. So for us, it's called the myelin sheath, which is right here. It's called Schwann cells in the PNS and oligodendrocytes in the CNS. That's not important to remember. But the point is it insulates our axon, and that makes the signal travel, like I guess you could say, better and more efficiently. So that's the function of that. But as you can see, the myelin sheath isn't everywhere, right? There's these like small openings, which are called nodes of Ranvier. And what they allow it to do is they allow polarizations to keep happening. So sodium needs to flow in from somewhere to get that like polarization stuff I talked about before, right? So these nodes of Ranvier allowed that. And this is called saltatory conduction, not because like sodium and potassium are salts, but it derives from saltare in Latin, which means to leap. So you can think of the signal as leaping from node to node. That's not actually what happens. It's more like the signal is propagating here, and then there's more like sodium, you get another depolarization, signal propagates, you get more sodium, more depolarization, etc., etc. So when I say a signal goes from an axon of one to the dendrite of another, what do I actually mean by that? So the way you can do this, there's two ways. You can either, they're actually like connected together, and you can have like, it's something called a gap junction. So the electrical signal travels directly, but that usually doesn't happen. Instead, we get something called neurotransmitters. So every time, the signal, let's say the signal reaches the end, right? This is the axon terminal, or sometimes called the axon button. But let's say it reaches the end, then it signals the neuron cell to release neurotransmitters from vesicles. And what these neurotransmitters, you can see it in red right here, they connect to these channels, and these channels will open it up and actually allow the sodium to flow. So remember I was talking about that depolarization stuff and that sodium flowing in and depolarizing the cell. So what does that actually mean? Well, it means that the neurotransmitter is connecting right here it's called a ligand-gated ion channel. Ligand-gated just means something like a molecule connects to it and opens it. But it actually connects to these channels and allows the sodium to flow in. So there's two types of ways this can actually influence the next cell. So in the postsynaptic membrane, like this cell right here. So either you can have, it's called ionotropic or metabotropic effects. Nothing too fancy. This just basically does short-term effects, and this is long-term effects. So this could actually change like gene expression and things like that. It can have effects lasting for weeks, whereas this is like a few seconds or a few minutes. So the response can be two types. It can be either inhibitory or excitatory. So in this example, the neurotransmitter opened the channel, but just as easily the neurotransmitter could have closed it, and that would be an inhibitory response. So I just want to make it very clear. Inhibitory doesn't mean that, the information is useless, or inhibitory doesn't mean that that stuff doesn't go to the brain. So I'll give you an example. Let's say in our ears, when you're in a car and it accelerates really fast, and your ears sense that acceleration, what actually happens is they send inhibitory signals to the brain, right? So that inhibitory signal is what allows your brain to detect, hey, you're accelerating, right? So it's not only that, excitatory signals are the stars of the show. This should be pretty intuitive, but let's say you have, two dendrites and then this is like the axon coming into these dendrites. So, if you have two excitatory signals, of course, but they don't reach the threshold and they're both like very far apart, then nothing will happen. If they're close together, then they'll, create an action potential, because like, let's say this one fires, but it couldn't quite get it over, but then this one kind of helps it get over the potential, the what's called threshold. So then you get an action potential. Spatial summation, basically you're just saying if they're closer together, you have more of an effect. And then this is inhibitory. So if one's excitatory and one's inhibitory, the cell might not fire. So one important neurotransmitter, for example, is acetylcholine, which is usually excitatory, but except for the heart, that's a special case where it's inhibitory. So usually what happens is, let's say acetylcholine goes out of the neurotransmitter, right? I'm sorry, of the presynaptic neuron. So this one right here. So the acetylcholine goes out. It's in this space called the synaptic cleft, and it's just lying there, right? Activating channels. But we don't want that to happen forever, like the response has got to stop. So then either the acetylcholine can go back, which is called reuptake, or it can get degraded by enzymes, which is what you're seeing here. So a more important example, you might have heard of the drug called Prozac. This is an antidepressant medication, and that's something called a selective serotonin reuptake inhibitor. So let's say this was not acetylcholine. Pretend it was serotonin, which is another neurotransmitter. So instead, serotonin is responsible for good mood and things like that. So you want more of it in your the synaptic cleft, because that promotes good mood in your brain. So basically what happens is usually it'll get degraded or something, or get reuptaken, right? But instead, what this drug does is it prevents that. So it keeps the serotonin kind of still lingering along in the synaptic cleft. So we have more of it here. And this is used to fight depression and things like that. So these are the types of neurotransmitters. You can have amino acids, you can have biogenic amines, you can have neuropeptides. This isn't as important to us. I just wanted to give you kind of like an overview that there are multiple types. They're not all the same. But this is more of like the biological focus on neuroscience. So the reflex. Remember I was saying in integration that stuff goes to our brain? Well, that was kind of a lie, because that doesn't always happen. So a reflex is the body's automatic response to the stimulus. We do it without even thinking. And so, for example, when the doctor uses a mallet to trigger the knee-jerk reflex, he's checking if your knee-jerk reflex is working well. So what happens here, we have something called stretch receptors. So when you strike the mallet, right, this tendon kind of gets stretched, and that sends a signal to our spinal cord, and the spinal cord kind of bypasses the brain. So usually this would go up and contact the brain would make a decision. The spinal cord is like, we don't got time for that. So then it just directly goes to this to the quadricep, and then this contracts, and then the foot goes up. So glial cells I was talking about, there's many types of them. These are just 4. So we have astrocytes, which are the blood-brain barrier. So that's why not all drugs can get to the brain. So when you manufacture drugs, you actually need to make sure that it can cross the blood-brain barrier. And these are what form the blood-brain barrier early in development. You have ependymal cells. This circulates the cerebrospinal fluid in the ventricle. The ventricle is an opening in the spinal cord and the brain. We'll talk about that more next lecture. And then we have Schwann cells and oligodendrocytes, which I already talked about. They have the same function. They basically allow the signal to propagate more effectively. And then we have microglial cells, which are very important because they're for immune function, right? So your brain is very sensitive to pathogens, more so than the rest of your body. So you need to make sure, your body needs to make sure that the brain is well protected from parasites. So one thing I want to talk about is you have 2 types of responses, right? You have automatic responses, and then you have kind of conscious responses, right? So the classic example, we'll take fight or flight, right? That is not a conscious response. You don't look at, let's say, a tiger running towards you, think about it, and shut down your digestion and things like that. Your body does that automatically. Your adrenaline starts flowing. You've heard that phrase before. So that's really like an automatic response versus a motor response, which is, you lift your hand up or you're writing with your pencil or something like that. So fight or flight, again, the sympathetic division, this is the fight or flight response. So, it accelerates your heartbeat. So you get more blood flowing to your muscles, right? It inhibits the activity of the stomach and intestines, because who really cares about your digestion when you're trying to run from a predator? It dilates the pupils of your eyes, things like that. Versus this has the exact opposite function. So it slows the heart. This is kind of, I guess you could say, the relax part of the autonomic nervous system. And then the final thing I wanted to talk about was something called long-term potentiation. So in our brains, there's a form of learning called long-term potentiation, and that increases the strength of the synaptic transmission. So it involves something called glutamate receptors. That's not as important, the name specifically. But basically what happens is, if let's say there's this connection, right, and it keeps getting used over and over and over again, well, our bodies are pretty smart. It actually compensates so we get depolarized more easily. We don't need the same strength. So that's why you've heard of something called use it or lose it in your memory. So let's say you study for a test and then 3 weeks later, you've never applied that knowledge. Well, chances are you're going to forget a lot of stuff because your brain doesn't need to establish this long-term potentiation, right? Because versus if you keep studying that material over and over and over again, it establishes that long-term potentiation, and you have this stronger connection between the two neurons, right? And obviously this is a simplified diagram. It's not just two neurons doing all the work. There's like whole nerve networks. So this is kind of an insight into what we're going to be talking about next time. We're going to be talking about the brain structure, the function of each parts of our brain. This is actually really interesting. The interesting part, you can actually see what parts of your brain affect like what you do on a daily basis. We're going to be talking about structures like the reticular formation, the thalamus, which is the integration center of the brain, the hippocampus, which facilitates the LTP I was talking about, things like the suprachiasmatic nucleus, which affects your sleep, and then your somatosensory cortex and your motor cortex. So this is a diagram showing like how much of our brain, of these two areas of our brain, are dedicated to the somatosensory and motor cortex. So for example, our face is a lot, right? So in both of these. So for example, touch affects your face a lot more than, for example, your knee. You're a lot more sensitive to touch on your face than you are to your knee, which makes sense from an evolutionary standpoint if you think of it that way. Okay, so next time we're going to be talking about the second part of neuroanatomy. And just to make very clear, I don't expect that you remember all of this, not even most of this information. The essential parts you need to remember are the following. You have a neuron, which has a soma. It has input centers called the dendrite and an output called the axon. Okay, strengths are always the same. A depolarization will always be have the same, the action potential will always have the same maximum value, no matter where it is. The strength does not vary. And then bundles of networks allow communication within your brain and within your body. So those are like the high points. So I would encourage you, if you're interested, I will put a reference in the description so you can look at that. There are definitely some interesting parts of neuroanatomy, but for the club, the expectation is some basic stuff, because we really won't be hitting too much on it. Just so you kind of understand the context where we're coming from when you talk about things like firing rates and spike potentials and things like that. So I hope you learned something and from this video. I hope that helped, and I'll see you next time.